Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show, bringing another really awesome guest today involved in creating a better tomorrow for all of us. Today, we have the honor of being joined by Dr. Mitchell Lee, who is the Chief Executive Officer of Ora Biomedical, which is a really interesting biotech company. They're developing small molecule therapeutics with a focus of extending lifespan and delaying chronic illness by ultimately targeting underlying molecular mechanisms of aging. Dr. Lee's research interests are focused on identifying healthy aging therapeutics, ultimately understanding the connections between aging and age-related diseases, and ultimately investigating how some of these natural genetic variations modify disease and therapeutic effectiveness. He's earned awards for science communication, multiple NIH training grants, and in 2015 was awarded a Howard Hughes Medical Institute Gilliam Fellowship for Advanced Study. He's also extremely passionate in terms of mentoring and managing sort of the next generation of scientists during both his grad and his postdoctoral studies. He mentored over 45 trainee researchers, ranging from high schoolers on through grad students. And in recognition for all his mentorship, he was awarded the University of Washington Undergraduate Research Mentor Award. He's also the founding chair of the American Aging Association Trainee Chapter, served on the Executive Committee and Board of Directors of American Aging Association. He received his bachelor's of science in biology, a bachelor of arts in philosophy from Western Washington University. He stayed at Western Washington, completed his master's in biology, and then moved on to do his Ph.D. In experimental pathology from the University of Washington School of Medicine, and most recently completed a certificate in biotech project management from University of Washington and UCSD in 2022. We're honored to have him with us. A lot of really interesting topics and themes to get into today. Dr. Mitchell Lee, thank you so much for taking time to come on the show.
Hi, Ira. Great to be here.
It's great having you. I'm very much looking forward to getting into all of these topics. A lot of really interesting things to get into. Love to start off, though, as we typically do, just by handing you the floor for a couple minutes to tell a little bit more about your background, a little bit of everything from where you grew up to why you got into biology in the first place, and a little bit of what got you thinking about aging and longevity. I think that'd be a great way to start off.
Yeah, absolutely. So that was a great introduction. That was pretty comprehensive on my background. I've always been interested in science ever since I was very young, just kind of seeing life and being curious about how it worked. I come from a family where education was something that was instilled as a value, but my parents were also, neither of them graduated from high school. So they knew education was the pathway to move forward. They knew that they needed to inspire me to go into education and do something worthwhile there, but they didn't really have a playbook. So it was a long road for me to kind of figure that out myself. I got very interested in philosophy and thinking about life from that aspect very early on. And then I started to see the molecular connections and the logic that underwrites genetics and how that informs all manner of different things that we see happening with living systems. That's been a perennial fascination for me. and something that was really kind of most awoken in me when I thought about aging. So aging was always something that I just imagined being a stochastic process, something that didn't really have regulation that underpinned it, that, it was just something that happened to biological systems, not something that biological systems had a hand in shaping. To learn that there is this genetic hand that shapes how our longevity works was fascinating to me. That has really inspired the last 10 years of my research, focusing on aging, the connection between aging and diseases like cancer, neurodegenerative disease, and then thinking about how those can be modified by both natural genetic variation and through small molecules. So small molecules I see as holding the potential for a new revolution in human health. It's a revolution that is happening now. We're starting to put together that playbook for it as we speak. And in the next 10 to 20 years, I think we're going to see a very different world because of longevity medicine.
Absolutely. Completely agree 100% with you. So now spot on. Yeah, I mean, so before we get into Ora and Wormbot, I'd love to start off. I took a look at your dissertation. It's entitled "The Consequences of Mutator-Driven Mutagenesis and Analysis of Lifespan Extending Compounds Using Outgrowth Analysis and Replicative Lifespan in Saccharomyces cerevisiae." And I thought this was extremely interesting because in many ways it sort of mirrors what we're going to be talking about next in terms of what you're doing with C. elegans and Wormbot, in the sense that you started here with the platform thinking about, okay, how do we do this translational GeroScience thing? How do we modify these yeast and look at the genetic diversity? How do we bring in all sorts of interesting compounds from drugs to natural products? Talk a little bit about your dissertation, if you would, because I think this is really interesting because it parallels where we're going next. I'd love to hear a little of that time frame. It's pretty cool.
Yeah, absolutely. Yeah, the dissertation isn't much of a beach read, but it was really fascinating to go through and do the research. It was kind of two different projects is what I did. The first project focused on cancer biology and understanding how fundamental cancer biology drivers impact cellular lifespan. So cancer is really a disease of random mutagenesis and random mutations. Some of those mutations confer particular abilities onto the cells where they can start to grow and divide and do their own thing. And that's really how cancer comes about. That broad mutagenesis doesn't just constantly give them abilities. It actually undermines their fitness in these really fundamental ways, such that the things that drive cancer also end up impacting the lifespan of the cells that are in that tumor. So I was really trying to figure out what that balancing act looked like. And we were able to find some different points where you could load in mutations and see a repeating phenomenon. So something, it was really interesting to be able to see something reproducible when you're building in something so stochastic. It's a mutation wherever and whatever it's randomly laid down. But above certain thresholds, you could start to impact cellular lifespan reproducibly, regardless of how those mutations were being spread across the genome. So that was one part of it. I've always been very interested in how we translate insights into the biology of aging out into the world to make a better life for humans and other, any animal that we want to have live a longer, healthier life, really. And that brought me into small molecule tests and trying to figure out how do we drug aging. That part of my dissertation really focused on using an invertebrate system, so in that case, budding yeast, to identify compounds with particular molecular activities that are very likely associated with longevity interventions. So, everybody's kind of gold standard right now for a small molecule in longevity is rapamycin. It's an mTOR inhibitor. So this system we developed with yeast allowed us to identify other mTOR inhibitors in a very rapid manner. So we quickly were able to screen through things. We found a lot of things with reported mTOR inhibitory activity that didn't show up in the assay. And we're able to validate it across a couple of different things as well. But, that really did, lead me into what some of this next part of this story is, which is, how do you find these interventions? How do you start to identify them? How do we get at the best of the best?
Mhm, mhm. And yeah, let's talk a little bit about Wormbot before we then get into the company. And as you know, Matt Caberline was on the show about a year ago, and we mainly focused on the dog aging project. He talked briefly about Wormbot, but I'd love for you to um, walk us through this in a little more detail, because you have C. elegans, you have artificial intelligence, you have really complex robotics going on. Sort of a really nice and elegant system put together here to do exactly what you're just talking about in terms of the screening, but on a much larger scale. Take us a little bit into Wormbot, if you would.
Yeah, absolutely. So the Wormbot is the technology we use to do high throughput drug screening to find these new small molecules. All of this research spins out from Matt Caberline's lab at the University of Washington. So Matt has been a field leading geneticist in the biology of aging for over 20 years now. He was one of the central figures to identify mTOR signaling as a promising lifespan extending molecular pathway. And Matt's lab was always really different. I came into his lab in 2012 as a graduate student, and the dog aging project wasn't on the horizon at that point in time. It was a lab that did incredibly high throughput genetics of aging, and they did that in systems like budding yeast and in C. elegans. So these are animal and fungal models that you can do a lot of work to understand basic biology of aging. And one thing that a lot of people ask is, well, that's a yeast or a worm. How could it have any bearing on aging in a human system? And that's really because aging happens at the cellular and molecular level, and those factors that get destabilized are highly evolutionarily conserved. So the same breakdown associated with aging in a yeast cell is very similar to the age-associated breakdown in function that you see in a human cell, and everything in between. So Matt's lab was very hot on the trail trying to understand aging as comprehensively as possible in the yeast and in the worms. And that meant growing large teams, doing throughput redefining science, and really trying to push against how much one lab could produce. Matt kind of created the playbook for that by screening the yeast genome collection. So it's about 6,000 different yeast strains, and you can take out one gene that has been removed from each one of those different yeast strains, and you can look at lifespan. So you can ask, when you get rid of that gene, how does it impact the cellular lifespan? So Matt's lab did that comprehensive genome level analysis. So they were very, very cued into doing larger scale studies. Doing larger scale studies in a worm model was the next goal that they wanted to do beyond the yeast lifespan team. So that was something that was coming online over the years while I was in the lab. And Dr. Jason Pitt was really pivotal in getting the Wormbot robotics technology up and running, where we can do these large scale lifespan studies. But it's really all out of the context of the Caberline lab at that point in time, which had this incredible interest in doing this scaled discovery research.
Mm-hmm. Mm-hmm. And that now becomes sort of the basis of Ora Biomedical. I mean, this a very important platform, as you're just mentioning. And if I could just read sort of your mission, the company seeks to realize the full potential of healthy aging therapeutics, developing a new generation of broad use small molecule healthy aging interventions for humans, companion pets, and all other organisms where prolonged healthy survival is desired. Take us just a little into the background of setting up the company, sort of where, how it exists. I mean, you're the chief executive officer, but a little bit of this core message I think is important, because I know I've heard Matt talk about it. And I've read, some of the sort of the social media discussions that you've had over, in recent months, that, hey, we got some interesting interventions that we're all testing, whether it's the rapamycins, metformins, and there's a list there, but it's not a very long list. And if we want to find interventions, we got to go kind of broad here. Talk a little bit about sort of how you view sort of the current GeroScience or sort of Gero small molecule landscape, because I think we're all in the belief that it's kind of limited at this point in time, and if you really want to find sort of those diamonds in the rough, you got to do a lot more searching. So take us a little into the strategy, if you would.
Yeah, the landscape of small molecule longevity interventions right now, over having 50 years to go through and discover and find things, it's humbling. There's a lot that we haven't done. There's a lot of promise that we found. So things like rapamycin show the potential of this space. We've got these interventions that extend the healthy lifespan of every organism we've given it to, and in some cases, very impressively extending lifespan. In other cases, it shows disease-specific or even age-associated disease-independent efficacy. It's a brand new type of therapeutic, but we don't have enough of them. I liken it to, over the last 50 years, if we were mining chemical space, all of the gathered field has basically been digging with their hands and hand tools to mine through chemical space to find these interventions. And we've been largely digging on top of each other because somebody found something that looks good. So we want to dig for something that's kind of similar to that increases our likelihood of finding something good. We haven't done a lot of broad, unbiased discovery at high throughput. And that's exactly what the Wormbot does. So, if everybody in the field has been digging with their hands and with spades, we've built a backhoe to comb through and dig through chemical space at very high throughput. And that's going to allow us to find not just the best of the best for the aging targets that exist now, it's going to allow us to identify brand new molecular targets that we can then find aging interventions for. And then what I'm very excited for is the promise of combination interventions.
Yeah.
So we've got single interventions that target different mechanisms. How do we put these together to create a cocktail that will maximize our healthy lifespan? So there's so much work to be done. The promise is so clear, though.
Mm-hmm. And let's go into each of those a little deeper, and we can actually do the first two together. And so, if we look at sort of the what you were just mentioning, you have the what you call the normative aging models, where basically, hey, we're going to take this small molecule, we're going to look at the C. elegans, we're going to see if it lives longer, and then you got the mutants, which will allow you to further sort of dig down and understand mechanisms. And we had this little Twitter chat, I think it was a few weeks ago, about if we get something that extends the C. elegans lifespan 3 times, but we don't know a mechanism, is that a bad or a good thing? It comes back to sort of the sort of way sort of pharma sometimes looks to this stuff, maybe a little too much on mechanism versus something actually doing something, a mode. Talk about these two parts of the system, normative aging models and disease mutants, working in synergy in your research program.
Yeah, absolutely. So we start with a wild type standard aging model. These are the same nematodes that have been cultured in research labs for decades. And we do standard aging studies. So put these populations of these animals in the presence of a given drug or drug combination, and then we just monitor their survival through their entire lives. So we can look at things related to lifespan and also some really sophisticated health span measures as well. Once we have those in a wild type organism, we can figure out a couple different things. We can get into mechanism if this is a small molecule that's unknown. So you can get different genetic mutants that have breakages in different genetic pathways associated with aging, where if the drug works in the background of that mutant, it's not working through that pathway. So you found a pathway independent from that important one. That's some of the main stuff you can do. You can do a lot of these are called epistasis studies with these genetic mutants. So getting into the mechanism, that's one thing you can do. The other thing you can start to do is figure out translational pathways. So, these interventions are not just going to be helpful in improving our long-term survival. They're going to have efficacy across numerous different disease states. So we actually have a panel of different age-associated and rare disease mutants that we test our hits across. We find for a given intervention, we find the disease state that it has the best efficacy for, and then that can provide us these pathways to move forward into mammal studies, into phase trials, and really figure out the quickest pathway to deploy these interventions to get them out to people who need them.
And the section on drug synergy, and as you mentioned before, is really quite interesting because I think that the HIV folks and the cancer folks have shown us there's probably not going to be a single magic bullet here, that we're going to have to understand, these combinations. And I think something interesting about your approach, I had a couple months ago at Aubrey de Grey on, and he was talking a little bit about what he's doing in terms of combinations, starting with sort of mechanistic stuff, and says, here's the senolytic and here's the mTOR inhibitor, blah, 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 and we're going to put that stuff together. In this particular case, it seems like you could go much broader, that maybe you don't need to start with these mechanisms. If there's A, B, C, R, and Q that do a great job, hey, there's an awesome combination, and we don't really care up front. We might do experiments later on and peel one away and see if the efficacy, but you can go a lot broader in the synergies. And I'd just love to, if you could tell us a little bit, maybe if it's confidential, we don't have to go into that, but how is a combination strategy sort of limited? I mean, if I wanted to test 20 substances versus 3, or what's sort of the strategy? Or because you can really, as you know, you can go pretty broad when you get into the synergistic combinations.
Yeah, there are problems of scale associated with doing combination studies, right, and doing these kind of fundamental dose response studies, right, to really get at the right kind of dosages. Right. So you've got to kind of play around in the space a little bit. You've got to have a tool that allows you to do a lot of these tests and different combinations in this kind of scale. So the Wormbot lets us do that. We can look at up to 120 populations in a single run, yep, and we can do upwards of 5 to 10 runs per bot per day, and we've got multiple bots running. So we've actually got this system where we can start getting into combination space. It's still not at that level of being systematic where you're just listing out every combination and going through it, but it gets us on a pathway where we can screen a lot more. So with the combination strategy we have in place right now, we take known longevity interventions, we validate those in-house, do that dose response to find the best dosages for those interventions. And then as we're finding new interventions, we test those across the panel of existing interventions to find those combinations. So we're actively, for every new intervention we identify, figuring out how it fits into this combinatorial longevity intervention space.
Excellent, excellent. And now another important piece of the strategy was, well, maybe not that recently announced, but is your million molecule challenge. And you recently wrote a paper in GeroScience in July entitled "The Million Molecule Challenge: A Moonshot Project to Rapidly Advance Longevity Intervention Discovery." And here again, I think this is quite brilliant, but here we have the ability, everyone listening here, to sponsor compounds, sponsor combinations of compounds, to throw into what you referred to as sort of this random intervention machine. Talk a little bit about the way this is set up. I sort of went into it a little bit before, and again, you said there's a mainly FDA approved drugs in there currently, so I could select whatever it may be and test things out and take a little time and go to the million molecule challenge, because again, I think it's quite elegant and exciting for sort of citizen science to get involved.
Great. Yeah. So, we have these Wormbots. We've got this scaled means of doing drug discovery. Since we've launched our independent operations at Ora Biomedical, we've actually made some really great innovations to our system to scale it even more. So very quickly, we're seeing that we've created a machine that can do something at a throughput that nobody is starting to scratch that surface. So that led to kind of defining this million molecule challenge. What is the most ambitious goal that we can achieve within 5 years if we had, the right amount of resources and the right amount of dedication to just doing the work. As a field, we just need to do the work. We need to turn the crank, find the interventions, do the combination studies in an animal system, and then start from a basis of knowledge that we can translate. So that's really what the Million Molecule Challenge became for us, is this central goal that we have to screen through 1 million interventions within 5 years. And so we've been talking about this, as you mentioned. We put out a paper on it, getting people excited about it. And, one thing that we always heard was, people wanting to get involved in some way, wanting to either, throw some money at it to donate towards it because it's a great cause, or to, sponsor screening through a library and a drug test with those, a lot of different ways to get involved. So we were thinking about how we really made a system of that. And that's how sponsorships came online. So we launched this just over a month ago, where you can go to the orabiomedical.com website. You can go to sponsor the Million Molecule Challenge, and then you can actually select longevity interventions that we'll test. You pay a small fee. We do the experiments for you, and then we get the data back to you as soon as the experiments wrap up. So it's really kind of quickly turned into this interesting citizen science approach that we didn't totally envision when we first thought about it. We saw that there was a lot of enthusiasm from people, but we never really imagined that it would be a way to engage anyone, anywhere, anywhere. If you have an idea for a test, we've got the time for it. Go in there, select the molecule, pay the fee. We'll get you data back. We'll be your scientific team. And so that has, resonated a bit. We've had a lot of people throughout the United States sponsor compounds. We're up to about 18 different countries who have individuals within those countries who have sponsored compounds. And we're just continuing to generate this enthusiasm around it.
And those, I know I asked this before, I think, or maybe I missed it. There, at this point, you can only sponsor a current FDA-approved drug, correct?
Yeah, we've created kind of a sandbox, essentially, where, what is the set of molecules that we can offer to people at the lowest rates? Yeah. So we needed to get a library and a system in place that we knew how to work with it really well, that was easy for us to, figure out how to scale it. So it's an FDA compound library that has about another 200 or 300 natural products in it. So yeah, it's a good combination of a lot of different longevity relevant small molecules in this format that allows us to basically take it to people where they can do experiments at this low cost.
Got it. Awesome. Something else I wanted to ask about as we're talking about synergy. And I know, going back to your PhD for a moment, I noticed that, one of the substances at the time that you tested was something, an extract called Pterocarpus marsupium, which it had some other bioactive substances in it. But again, going back to the area of natural products, when some of these things, these interventions, like plant extracts, are kept in their natural form versus sort of the old, the pharma method of purifying something out of them, they're little combinatorial libraries of substances. How do some of those potentially impact? Are there issues? I know plant extracts were always an issue in sort of traditional cellular screens. There's some benefits that sort of Wormbot opens up for allowing us to test, because there's literally, I was back, I'm thinking back to my ag tech days. There was something like, 300,000 species of higher plant on this planet, very few which we'd ever really looked at in depth in terms of the combination bioactivities that they have. Any interesting insights there on how Wormbot can help with that screening as well?
Yeah, the Pterocarpus marsupium story kind of gives us some preliminary insights into how we would go around kind of better investigating those extracts. So
Right.
yeah, this was something that we tested in a yeast system. We found that it increased the yeast's replicative lifespan with treatment.
Mm.
It was a finicky extract. It was
Oh, yeah.
actually something that showed a tremendous amount of light sensitivity. So we
Mm.
had to keep the plates shielded and do everything that we did as much as possible in the dark with it. Very likely that is because of one compound in the extract called pterostilbene.
Mm.
Pterostilbene, it's actually related to resveratrol, which for
Right.
people in the longevity scene, that's an infamous molecule by this point in time.
Right.
But
Right.
pterostilbene is one that's thought to have a greater bioavailability than resveratrol, but it's got this photoconversion problem. So we were looking at that. We were seeing that relevant doses of pterostilbene were not reproducibly getting us the lifespan extending effect, but we were seeing it off and on, where it looked like, it was kind of a technical thing that was making it such that we weren't seeing it when we weren't seeing it. So that can give you some insights into it. There's a lot of great mass spec that's been done with these extracts. You get information on the extract, you figure out what is in those extracts and thought to have the bio properties that it has, and then test those in independent and test those in independent combinations outside of the extract to
Yeah.
see how closely you can recapitulate that impact.
Yeah, and I'm just thinking back to my days with natural products, and yeah, they're finicky to work with. But again, if there's again, interesting systems like what you're developing here to allow us to take another look at them, and that then gets me one more area. I just love to hear your feedback on because I know that in this was November 2021 in Science, you wrote this piece called "Anti-Aging Diets: Separating Fact from Fiction." In this case, you were talking about how we really don't know about how a lot of these dietary interventions work, and in this case, you're proposing in certain cases there may be mTOR focused pharmacology in these foods. But again, thinking of that tremendous mass of plants out there, and something like 50,000 of them or so are edible. We only eat a few in our diet. I see there's some possibility here for sort of this food as medicine, stuff that we see happening. I just actually a show on that a couple weeks ago with a major supermarket chain here in the United States. Any plans? I mean, probably too early if it's confidential. We don't have to go into it. But any plans for sort of again looking at sort of the medical food aspect of what you can learn from something like combining Wormbot with these other non-pharmaceutical interventions that may do things like hit mTOR?
Yeah, absolutely. We're engaging that in a couple of different ways. One way is to, we might find lifespan extending interventions out of this, but we want to do a big study of all of the grass compounds, so generally
Yep,
regarded as safe. So
excellent.
one part of that is to, see if there's anything on that list that has pro-longevity outcomes. The other thing is to see if there's anything that has a negative impact on longevity when you look at it longitudinally in an animal. And that's something, I think there was something about a red dye or something here lately that was on the grass list, and then they're having to take it off, and it's been listed as a carcinogen by California, I think is what it is.
Yeah.
So yeah, doing that kind of thing and just saying, okay, well, aside from an Ames test or some kind of mutagenicity test or something like that for a compound, can we learn things about their safety by looking at an animal system exposed to it longitudinally? So that's one side of it. The other side is we're always excited to get our hands on different extract and different natural product libraries. There's some people who have, some great plant libraries out there.
Yeah.
One thing that I don't think has been researched enough are marine extracts.
Oh, yeah.
So yeah, I'd be super interested for us to do a large screen through the marine chemical space.
Yeah. Yeah, that was I remember that was always one of the areas that even back in the day when we were exploring the plant world that yeah, no one was touching. And a long time ago when I was at SmithKline Beach, and we actually had scuba divers that went and looked for things in the ocean. So yeah, no, it's interesting, seeing sort of the reconnection of this stuff that, I think most of the smart people tell you we've not scratched the surface on a lot of this biodiversity. And if we can put it into the system with these new technologies, I think that's going to be awesome. So I'm excited for what you're up to. What else is coming up, Mitch? There in terms of the company, if there's as we're getting into 2024, I mean, I know obviously you're doing a lot of this. You have the Million Molecule Challenge. You're doing a lot of this also as a service. But what is sort of some of your holy grail dreams? I mean, if I, $1 billion dropped out of the sky tomorrow into your bank account, where do you want to take some of this? I mean, what is your goal? Is it to do the dog aging project part 2 with some of these moieties? Is it to use some of these FDA approved compounds and think about combination therapies in humans as soon as possible? Where do you want to go with all of it?
Yeah, yeah. So in terms of our studies, the first thing is to do the work. We want to comb through chemical space, find the very best of the best interventions, the combination interventions. How we deploy those, I see a couple different strategies starting to resolve themselves. One is going into larger mammals like companion pets. So, some of the announcements that's been made by Loyal over the last week showing how they're starting to move forward longevity medicine itself through the FDA is incredibly promising. And I think that's a great strategy to move forward with. Whether we move forward and develop our own therapeutics or we work with people who are doing those clinical trials and do that partnership there and out-license our interventions is kind of an open question. We see ourselves having the problem of having too many great interventions to move forward. So how do we deploy those in ways that basically get them out to the people and pets and animals who need them as quick as possible? So out licensing is one thing. This is kind of more in my soapbox territory. But as we think about clinical trials in GeroScience, right now we've done a lot to select our populations based on chronologically aged but healthy people.
Right.
And that's great. I wonder, however, whether we're missing the opportunity to help people directly today. What you see in the lab is across genetic variation within a species, the shortest lived organisms are the ones that reap the biggest benefit from longevity interventions. Right. And, you see, this kind of survival stratification in human populations as well. It stratifies based on sex and race and socioeconomic status. So are there ways that we can utilize and leverage longevity medicine today to help out some of those people in those shorter lived demographics? Mm-hmm. That's something that I think, more and more of my time, I'm thinking about how we can actually help people quicker in those groups.
Yeah, it's an exciting time, and the fact that you're showing up with again a really unique and elegant way to expand the size of needles in the haystack, which we definitely need, is just is so refreshing and important. Anything else I missed, Mitch, as we get to 2024, anything coming up that you want to mention, the public facing stuff, conferences you're going to be talking at, anywhere we can run into you, meet you, anything else that I missed, please take the floor.
Yeah. So we're going to be making a couple big announcements probably by the end of the year regarding sponsorship and partnerships with the Million Molecule Challenge. So stay tuned there. So we're going to be continuing to grow that, and we've got some really cool partnerships that we're starting to put together with that. In terms of next meetings for me, I'm kind of a perennial figure at the American Aging Association meetings. So the national meeting this year is in Wisconsin. I want to say it's 1st of June. That is a great meeting if you want to learn about the science of aging and interact with a lot of the people on the ground floor doing the work. That's something that has been a little disappointing to me, is how little cross-pollination there are between a lot of the people who are excited about the space and a lot of the people funding the space through venture and different kind of capital, and the scientists who are, quietly in their labs doing the work and making the discoveries. We need to have more interaction between these different stakeholders.
I agree with you 1,000% on that one, and I'll let that one. We'll talk about that one on the next episode. But really, really awesome stuff, which I look forward to continue to follow what you're up to. I'll probably throw some money in and sponsor a few compounds myself. I have some ideas, but yeah, really great stuff. Again, for everybody that's going to be listening to this particular episode of our show across the various podcast networks, or watching on our YouTube channel. Again, you've been spending time with Dr. Mitchell Lee, Chief Executive Officer, Ora Biomedical, doing really amazing things to discover and develop these novel small molecule therapeutics to ultimately extend lifespan, delay chronic illnesses. Mitchell, I want to thank you again for taking the time out of your schedule to come talk to us, to educate us on all the exciting stuff you're up to. Obviously, thank you for what you do. And as we like to say on our show, thanks for helping to create the better tomorrow for all of us via the work you do. Really great story.
Awesome. Thanks, Howard. Great to be here today.